Thank you for watching part 2 of the Biennial Bearing in Apples on-demand webinar series. In this session, you will hear Professor Jens Winsche and Anton Milyayev from Hohenheim University and Dr. Henrik Flarowski, Director of Fruit Breeding Institute in Dresden, Germany, share their research outcomes about the mechanisms involved in biennial bearing and why apple crops fail to develop sufficient flower numbers in the year after a high crop load. This Hort Innovation project is a collaboration between Agriculture Victoria and the University of Hohenheim in Germany and is funded using the Apple and Pear Levy. To view part 1, please visit the video and webinars page on the APAL website. If you have any questions for the speakers, please contact Rose Daniel, the Technical Manager at APAL.
Okay, the project is trying to find the underlying mechanism of biennial bearing in apple. There are two parts to it. We did some physiological work and some genetic work. And the presenters are going to be Anton Milyayev. He is a PhD student at Hohenheim University. Henrik Flarowski, he's a director of the Fruit Breeding Institute in Dresden-Pillnitz, and myself, I'm the chair of the Department of Crop Physiology at Hohenheim University. This is just briefly our outline, what we're going to present. I'm going to discuss very briefly what we understand under biennial bearing in apple. I will also discuss the experimental design. Anton will follow on discussing the histology we have done in this study. And he will also present results on proteomics and metabolomic profiling. And Henrik Flarowski, he will discuss RNA sequencing, the gene work, and he will present an outlook what comes next. Well, biennial bearing in apple is the annual cycling of on and off trees. You see here on this slide, on trees, they have a very high flower density in spring. This sets up a very high fruit load, but in that particular year, very few spurs are going to be initiated. Buds for the coming year are not set up, and this is taking the tree into a off year in the following year. There are no flowers in the year to come. In contrast, off years, they have few or no flowers, consequently no fruit load on the tree. But in this particular year, very many buds are going to be initiated, setting up a very high flower density in the following year. Here, I show you briefly the experimental design. We have the contrasting trees in the slide, in the picture, at full bloom and at harvest, on and off trees. We worked with two different cultivars, one Fuji, which is a highly susceptible cultivar to biennial bearing, and Royal Gala, a very regular cropping apple variety. We have two treatments, on and off, as I explained before, and we sampled the buds on 2-year-old wood, spur wood, over 15 weeks, starting 4 weeks after full bloom. This is the experimental design I already discussed in the previous slide. Also the sampling. What did we do with all the buds we sampled? Over 2 years, we sampled approximately 30,000 buds. And this was necessary to undertake all the various measurements. Some buds we used for histological work. We wanted to determine flower initiation, when it exactly occurs in time, and we did some omics analysis. What do we understand under omics? Well, we first looked at the genome. This is all the genes coding for specific proteins. And we looked at also non-coding genes. This DNA, the desoxyribonucleic acid, is transcribed into RNA, ribonucleic acids. And this is the building block, the building blocks for proteins, which we evaluated in detail, the structural and non-structural proteins. And again, those proteins are the building blocks for metabolites. And we were particularly interested in carbohydrates, in amino acids, but also in phytohormones. And we looked at the whole proteome and metabolome in those particular buds we sampled. So I'm going to hand over to Anton.
Thank you, Professor Wünsche. The very first step to study biennial bearing in apple was histological sectioning. Histological sectioning shows the stages of development of each and single bud that we collected in 2 growing seasons, in 2015 and 2016. Every bud undergoes the whole developmental process consisting of 2 different stages. The first 2 stages are considered to be vegetative. As you can see on the picture, there is a small tissue marked in red. This is a flower meristem that is developing here in the middle of the bud. And then starting from the stage 3, we can already see the growth and expansion of the meristem. And at the stage 5, we can observe in the bud already small flowers, which will come out next year. If we look at the results, we can see on the vertical axis the same 5 stages of bud development. And in Fuji on trees, in trees with crop load, we can barely see flower initiation. Just 1 bud was found to be initiated in 2015. In contrast, in Fuji off trees, we can see an extensive flower bud initiation. It started 75 days after full bloom, and then every week we could find a bud with small flowers in it, as it is depicted in the upper right corner. Then all the data was transformed into binary data, and we created logistic regression model from this. The logistic regression model predicts the onset of flower bud initiation that is defined as 20% of the maximum initiation rate and marked in arrows on the graph. The predicted flower bud initiation occurred even earlier than microscopically observed, but this model logistic regression model needs to be validated with the following experiments and with the buds we received from Australia to confirm it. What do we have? We have time points of flower bud initiation for both cultures, for Fuji and for Gala, and in both years, 2015 and 2016. But what does it mean for growers? That means that if the trees are remained with high crop load until this time point, then the flower initiation of these trees will be strongly suppressed and the trees will fall into biennial bearing. But what does it mean for scientists? That means that these weeks prior to flower bud initiation are essential to perform comprehensive analysis, such as gene expression analysis or RNA sequencing, analysis of proteins, and analysis of small molecules, such as sugars and flavonoids, which may serve as signaling molecules for flower induction. Our proteomic profiling showed that in spur buds, around 7,000 unique proteins could be found. And all these 7,000 proteins underwent the statistical and quality filtering. After this filtering, around 300 proteins remained in the data set. Some of them could be linked to the differentially expressed genes between on and off trees that makes these proteins even more interesting to look at. What do we have right now? We do have a proteomic profile of each stage of flower bud development, and the most interesting proteins are to be involved in the metabolic pathways promoting or inhibiting flower initiation. And we continue studying these proteins right now. Metabolic profiling. During the metabolic profiling, we could identify small molecules, compounds from the groups listed on the right side of the slide. What are the key findings of metabolomics? We detected chlorogenic acid. This compound was more abundant in spur buds collected from off trees compared to on trees, with prominent cultivar differences. It was approximately 5-fold higher abundant in Gala compared to Fuji. And tryptophan, this compound showed an opposite pattern of abundance and had even higher cultivar differences and approximately 10-fold higher abundant in Fuji compared to Gala. But what links all these compounds together? Chlorogenic acid has in plants a plant hormone-like activity, in particular in auxin activity, and tryptophan is a precursor for auxin biosynthesis. That means that plant hormones should be actively involved in flower bud initiation and as well as in flower bud induction. So what we plan next, we are aiming to analyze the profile of phytohormones and sugar molecules with potential signaling activity. By that, I mean all the detective sugars and all detective phytohormones. We will use these compounds as potential compounds for external application for apple trees to manipulate biennial bearing. In the next part of the presentation, we will present the results of our genetical analysis, and I'm giving word to Professor Henrik Flachowski. Thank you.
Yes. So, as Jens told you before, we were not only interested in proteins and metabolites, which are differentially expressed between on and off. So we were also interested to find the genes which are differentially expressed between the two trees and which are the reason for in the bearing behavior of Fuji and Gala. On and off trees, we isolated the RNA and then we enriched the mRNA to sequence all the transcripts of genes which are differentially expressed. The results of our investigation can be seen on the left-hand side. RNA sequencing resulted in more than 9,000 genes differentially expressed between on and off trees. Out of these 9,000 genes, 5,910 genes were only differentially expressed between on and off trees in Fuji, whereas 2,369 genes were differentially expressed between on and off trees in Gala only. 1,057 genes were differentially expressed in both cultivars. This is interesting because we were interested to see which genes are differentially expressed between on and off in both varieties, because this gives us an idea about the common mechanism underlying biennial bearing. More interesting for us are genes which are differentially expressed only in Fuji or only in Gala, because this will give us an idea what is the difference in the bearing behavior between a variety like Fuji, which is susceptible to biennial bearing, and a variety like Gala, which is regular cropping. After sequencing all the 9,000 genes, we tried to assign all these genes to different plant pathways, to metabolic pathways, but also to general pathways. On the right-hand side, you can see our results showing genes which are differentially expressed at 55 days after full bloom and 63 days after full bloom. These genes could be assigned in Fuji off trees, for example, to biosynthesis of unsaturated fatty acids, to fatty acid biosynthesis, but also to DNA replication, to photosynthesis, or to biosynthesis of amino acids, for example. In Fujian trees, we found genes which could be assigned to plant hormone signal transduction, or to plant pathogen interaction, or to phenylpropanoid biosynthesis pathway. So using this information, we tried to establish a first model of the genetic mechanisms underlying biennial bearing. From literature, we know that there are still different hypotheses about the underlying mechanisms. It is common knowledge that the genetic makeup of a cultivar has a strong impact onto the bearing behavior. As Jens told you, some cultivars like Fuji or Elstar are known to be susceptible to biennial bearing, whereas other cultivars like Gala, for example, or Pinova are regular cropping. Beside the bearing behavior of a cultivar, plant hormones produced by developing seeds seem also to inhibit flower induction in nearby meristems. We also know that carbohydrate limitations, the competition for carbohydrates between the developing fruit and nearby meristems, are also discussed as possible reasons for biennial bearing. Currently, it is not really clear if one of these mechanisms is the most important one, or if these mechanisms act in concert together. Based on our results, we suggest the following multi-level concept for a flower bud initiation in apple. The first level is determined by the genetic makeup of the cultivar. The genetic constitution of the plant has a direct impact onto the onset of flower initiation. As I told you before, some cultivars based on their genetic makeup are biennial bearing, whereas other varieties are more or less regular cropping. However, the genetic makeup also affects crop load, and crop load directly affects the onset of flower initiation for the following year, and therewith the bearing behavior of the cultivar. Based on our model, crop load is a second level determinant for the onset of flower initiation. During the season, the bud accumulates temperature, accumulates heat And heat is known to have a strong impact onto floral initiation too. In our model, heat accumulation is a third level determinant for the onset of flower initiation. So what do we want to do in the future? For the moment, we have huge data sets for metabolites, for proteins, and for genes. What we are still missing is information about plant hormones and different sugars and sugar forms, which are also expressed in apple buds and fruits. Currently, we are trying to identify those hormones and sugars in the buds we collected. We are working on the selection and the validation of candidate genes for flower induction, and using this information, we try to validate our model for the prediction of flower initiation by studying apple trees kept under controlled climatic conditions. We are further working on the identification of metabolites which promote flower induction and which are suitable for foliar application in orchards. And our final goal is to identify genes underlying the genetic mechanisms of biennial bearing, and based on the differences we can find in these genes between regular cropping varieties and biennial bearing varieties, we can establish molecular markers for selection to be able to breed new varieties which are less susceptible to biennial bearing for the future. So we would like to thank Horticulture Innovation Australia and the Deutsche Forschungsgemeinschaft for financial support of our research projects. We would also like to thank our collaborators from the Competence Center of Fruit Growing Lake Constance, from the Julius Kuhn Institute, and also our French colleagues from INRA, then our colleague Dario Stefanelli from Agriculture Victoria, our colleagues from Italy, from the Edmund Mach Foundation in San Michele alla Dice in South Tyrolia, but also the colleagues from CUBIC, this is the Genomics Center of University of Tübingen, and the colleagues involved in this project from University of Hohenheim, Jens Wünsche, Jens van Stiel, and Anton and his colleague Julian Kofel. I would like to thank you for your attention, and if you have any questions, don't hesitate to contact us. Thank you very much.